Hello everyone, I made this video to provide you with the answers to the reassessment that we just took. So the first question, I asked you to draw an acid-base curve for distribution group for cysteine. So I was trying to do this quickly, so I didn't draw my own structures. I got these from the internet. So the pKa's aren't quite exactly the same, but you get the idea. Cysteine, when it's fully protonated, has a sulfhydryl group, an amino group, and a carboxylate group. And the pKa of the least held hydrogen is the carboxylate group. So here it's 2.05. So it's about 2. But as you progress from a very low pH to a higher pH, this is the first proton that will come off. So here we have 3 ionizable protons, and if we add up the charges, that has a +1 charge. If we take off the first proton at the pH of about 2, we see that we have a negative charge here and a positive charge here. So we still have our 2 dissociable protons, but it's not charged. So that's our zwitterion right there. The next proton to come off is the sulfhydryl group, because we go from 1.92 to 8.37, or about 8 over here. So the sulfhydryl group comes off next, and when you take off that hydrogen, that becomes S-. So we have a plus and a minus and a minus, so that's HA-. And then the last proton to come off is the amino group, and it has a pKa of about 10-ish, and when that proton comes off, this is neutral, and these two are negative, so we have A2-. So when we plot these, our crossing points are going to be at half the formal concentration, 0.0025 molar, and we cross at 1.92, 8.37, and 10.70. And because our zwitterion is here, our PI is going to be halfway between 1.92 and 8.37, or it looks like it's about 6-ish. So the zwitterion is the point on the curve where the zwitterion concentration is the highest. So where that arrow is, that would be the PI. Okay, so the next question, we looked at one of the patients we looked at on our first exam. So that's Rashmi, and she had a beta globin chain where there was a glue to lyse substitution. So the wild type DNA would have a glutamate, and the lysine would have hers has a lysine. So if you look at the genetic code, because of the redundancy of the code, there's actually only one right answer to this. I'd have to go back to the paper and look, but you wouldn't have known that. So if you looked at the codons, you'd see it would be GAA or GAG, and that's for glutamate. And then for lysine, it's AAA or AAG. So I've circled the change right here. So in either case, there is a change from a G to an A in the first position of the codon. So I introduced another patient to our reassessment. This is Henrik. He's 37 years old. He has a high hematocrit, which means his red blood cell count is very high, high hemoglobin levels, and his physicians think he might be heterozygous, which means he inherited one mutant hemoglobin for one parent and one normal from another parent. So they collected samples from his blood and purified the hemoglobins. So they found out his alpha chain was normal, but there's a beta chain in which the lysine residue at position 144 was mutated to an asparagine. And they searched the literature, and it turned out that this hemoglobin had been isolated before, and it was named hemoglobin Andrew Minneapolis. There are other things about this patient that are interesting that we didn't have a chance to get to in this case study, but maybe some other time. So if you take the hemoglobins from a normal person, hemoglobin A, sickle cell hemoglobin, and I reminded you that for sickle cell, a glutamate at position 6 in the beta chain is mutated to a valine. And if you take hemoglobins from Henrik, then you should draw the isocyclic focusing bands. So just to remind you of what isocyclic focusing looks like, you will load proteins at the top of the gel where the pH is very high, and there are not very many protons, but as the pH decreases, the proteins will become protonated, and when you become protonated, they will get to a point where their isoelectric point is reached, where they are no longer charged, and they will no longer migrate in an electric field. So basic amino acids don't have to migrate there that far, but acidic amino acids do, because they have carboxylate groups that have a low affinity for protons, and it takes a lot of proton concentration which you find at a low pH, to get those COO minuses to go to neutral COOH. So to answer this question, I made myself a little table. So we have lysine at position 144 in normal hemoglobin and glutamate at beta 6. For hemoglobin, the sickle cell hemoglobin, the valine, there's a glutamate to a valine, but the lysine would still be there. And then for the hemoglobin Andrew Minneapolis, you'd have an asparagine, but the glutamate would still be there. So if you look at this, if you think of this, the and everything else is the same. So these are the only residues we need to look at. So if you look at hemoglobin A and just put that maybe in the middle of your isoelectric focusing gel, you can see the hemoglobin S is more positive because we get rid of a negatively charged glutamate. And we can see that the hemoglobin Andrew Minneapolis is more negative because we have a glutamate here, but we lose a positive charge. So that means that hemoglobin S will not travel as far, but the hemoglobin Andrew Minneapolis will travel farther. So this was what should look like. Here's hemoglobin A, and there's hemoglobin S. If we have hemoglobin from our subject, the hemoglobin Andrew Minneapolis is going to be down here, and we have two bands because our hemoglobin from our subject, he's heterozygous, so he has a normal hemoglobin from one parent, and then he inherited hemoglobin Andrew Minneapolis from the other parent. And I just wanted to show you, this is the actual gel from the paper. So they show hemoglobin A right here. And this is sickle cell. So I didn't give you this information, so I didn't expect you to have this. But they ran, this person is heterozygous for sickle cell. So they have a hemoglobin A band and a hemoglobin S band. And then there's the subject right there. So hemoglobin A, and there's the hemoglobin Andrew Minneapolis. And they also looked at another hemoglobin called hemoglobin Little Rock, which we didn't look at. But I thought you might find this interesting. So now what we need to do, because he's heterozygous, is we have to purify the hemoglobins from each other. So we have to separate hemoglobin A from hemoglobin Andrew Minneapolis. So we're going to use anion exchange. And remember, anion exchange is named not for the bead, because the bead is positive, as we show here, but for what binds to the bead. So negatively charged proteins will bind to the beads, and positively charged proteins will be repelled. So this is an example of the positively charged bead. So we can think of the positively charged molecules being repelled. They'll come off first, whereas the negatively charged proteins will be attracted and they'll come off second. So we already talked about how hemoglobin Andrew Minneapolis was more negative, so it's going to be attracted, so it'll come off second. So hemoglobin A comes off first, and hemoglobin Andrew Minneapolis comes off second. Some people drew hemoglobin S in here, but Henrik doesn't have that. Remember, we're looking at this subject, so we don't have a mixture of all of them. So there should have been a third peak in here. So now that we have the hemoglobin, we had the blood cells, we had both hemoglobins, we separated the hemoglobins, and now we're going to separate the alpha and the beta chains. Do a tryptic digest, and what kind of fragments are you going to get compared to normal? So I wanted to remind you that trypsin cleaves after arginine and lysine, but the cleavage doesn't occur if the proline residue comes after that. This is the sessile bond right here. So the way that this works is that if this amino acid up here is a lysine or an arginine, if N-1 is arginine or lysine, this bond will be hydrolyzed as long as N is not proline. So I know you didn't have this information, but I just thought it would be fun to give it to you. So here's the sequence of the hemoglobin A beta chain, and here's Andrew Minneapolis. So if I direct your attention over here, this K right there, that's on 144. So the beta chain has 146 amino acids, so it's right at the end. So instead of a lysine right here, you have an azine. So what happens is you get one fewer fragment. So instead of having two fragments right here, this fragment is not recognized. And I didn't give you this information either, but you should have addressed it. There isn't actually a proline that follows it. If there had been, then there wouldn't be a cleavage site here, and then your answer would have been different. But there is actually no proline here. So we do cleave at this lysine, and we don't cleave here at this azine. So the next question I asked you was, I gave you an oxygen binding curve. So we have millimeters of mercury, or torr, on the x-axis, and the percent oxygen saturation. So they have it percent. They have from 0 to 100% rather than 0 to 1. So y is just going to be fraction from 0 to 1. So so anyway, you determine the P50 from the graph. So you draw a line over at 50%, because that would be y equals 0.5. And P50 is that oxygen partial pressure at which y equals 0.5, or 50%. So if you drop a line down, and again, you're reading at this graph, so I get 30 and 17. Your values might have been slightly different because we are reading off the graph. We do have to have our units because P50 has the same units as oxygen partial pressure because of the definition, because P50 is that oxygen partial pressure at which y equals 0.5. So I looked at your values and looked to see if your y values that you're calculated were consistent. I had a little Excel spreadsheet that I pulled up. So again, not everyone's going to get the same answers, but you should have got something that's close. I didn't ask you this, but just this is kind of interesting that, so the propositus, that's Henrik. So if you look at his P50, it's very, very low. So, his oxygen, his hemoglobin has a high oxygen binding affinity. So you can see it doesn't release as much oxygen. And he's got less cooperativity too, because his N is lower. But that's just for interest. We didn't cover that on the reassessment. So here are my calculations. So I gave you the calculations. So for normal hemoglobin, we at 20 torr, I used my 30 torr as the P50 and the N value that was provided, and I get Y is 0.251, or 25% at 20 torr, and 0.934, or 93% at 80 torr. For our subject, again, his P50 value is a lot lower, and his N value is also lower, so his Y value is much higher, indicating a higher oxygen binding affinity. So his Y value is 0.596, and his Y value at 80 torr is 0.976. So anyway, those are the answers on the reassessment, the class did very well. Lots of points were gained. The class average went up by 14%, which is lots of good learning and growth mindset attitude. So congratulations on taking the time to go through this exercise. So I hope this was helpful. I'd be happy to answer any questions that you have in office hours. Thank you for your time and attention.